So we'll get started for the third WNT webinar. Today, the topic is scientific and test method readiness of emerging technologies, criteria, examples, and experience. So we will have today the following five speakers. As you know, this series of webinars is there to help the WNT get familiar with a number of topics that have been prioritized by the WNT itself. And this is all in preparation for our workshop, where we will discuss any necessary adaptations to the test guidelines program later this year in December. And so the webinar is recorded and the recording will be available on the WNT community site. Also the presentations will be all available on the WNT community site, as well as a short report that will be prepared by the Secretariat and the steering committee for the preparation of the workshop. So if you have interventions to make, you may use the chat box for raising questions to the speakers or to initiate some discussion also with other WNT members. So there won't be time for live discussions during this webinar itself. Okay, so we will have today as speakers Marcel Leist from the University of Contans in Germany will give us a talk about the experience gained in all the work on developmental neurotoxicity. Then Elise Grignard from Pepper platform in France will give us a presentation based on her experience with the platform where they do pre-validation of endocrine disruptor assays. Then Monique Perron from the US EPA will give us a talk from her experience with the new approach methods that are considered and the evaluation of their readiness level. And we will conclude with a presentation from Hajime Kojima from NIHS in Japan about his experience at JAGVAM with the validation of a large number of methods and what are the learnings in terms of evaluating the readiness level prior to the validation studies. So I just, in order to give a bit of maybe inspiration for questions that could be raised in the chat or food for further thinking. I listed here a few questions that I just came up with earlier today. Really thinking about this topic of readiness assessment from the scientific and technical point of view. So the one question could be whether the concept and the criteria associated with readiness are broadly applicable, or are they context and method specific? Are we thinking about some checkbox, some systematic criteria, some scoring versus more case-by-case and context of use dependent evaluation of readiness? And I think the speakers today will provide some insight on this in particular. Also, are there specific aspects of a method that are consistently insufficiently ready when they are presented? And that would be informative to guide us on whether we need to develop some specific guidance or communication. Is there existing resource and guidance for test developers to understand and self-evaluate when a method is ready for test guideline development? And if such guidance or publications exist, is it sufficiently communicated? And is the impact measured? Also, should time and resources be invested in better communication and training on these criteria and guidance for readiness? And also, you know, are we reaching the developers community sufficiently? Are they accessible? Are they receptive to the needs of the regulators? And should more efforts be made to reach that community? Where, who are we talking about? How should we communicate? How should we, you know, what should be communicated? Publications, guidance. So, yeah, these are just, you know, kickoff questions. There will be certainly more questions. Please use the chat box as you understood. And let's get started with our first speaker. So I'll stop sharing.
Welcome. And I'm happy to you, Anne, for this opportunity. And I think already now with Miriam, it will be a nice complementary talk to reinforce some elements and to add into some gaps. So just to tell you, I'm warming up a little bit with validation, then going into the readiness criteria, and I will mention another template, which Miriam mentioned as an Excel sheet, which we call ToxTemp. And then I will show two examples from a European project and from the EFSA project on DNT. So just to get everybody on one plate again. So of course we have these many tests, but for the regulatory acceptance, this is not the right line, but actually the regulators do have to want this test for the acceptance, and for that they need some trust. And we would say that the major feature, or the feature which is absolutely necessary, is reliability and reproducibility, and that will also be the main focus. Of course, there are other elements. There is, of course, the predictivity and the relevance, which go then also in the performance of the test, but that is kind of a second step. And as mentioned before, these two features, they are not only dependent on the test. So the reliability only depends on the test. The predictivity and relevance, of course, refers to a reference system. And in some areas, it's relatively easy to generate such a system and also to find reference chemicals. In other areas, it's incredibly difficult, for instance, in developmental neurotoxicity. So therefore, we are a bit more careful here. On the reliability, just it's like a basket of eggs. Everybody has their favorite feature there. It's also a terminology issue. Some call that robustness. And of course, there are all these features on how well the assay works. So I'm just giving some examples here, and of course, you will have the slides later. So the point is, everybody has their favorite egg, and some may be missing and some may be double. So the question is, how do we formalize the reliability criteria? And of course, I guess Hajime is going to much further here. So the note here is the target is not fully validated. So OECD standard assays, but the many advanced but not absolutely perfect test methods. So we talk an intermediate level. And this paper was mentioned before, so we went together with lots of people, so Magda from the OECD, Tim Schaefer from the US EPA, Andrea Perron from EFSA, and so on. And we thought, like, can we pick DNT as an example to set up such readiness criteria? And this is just to show that readiness means something different for different stakeholders. So for an academic investigator, it's if the assay works scientifically, it's fine. But that's not good enough for a toxicologist that wants to classify as positive or negative. And of course, a regulator also has different wishes towards readiness. So I'm talking about this intermediate level. So to have a robust and relatively relevant assay. And just to give you an overview, we defined 13 areas listed here as domains, and that could be the test system, the main endpoints, the control chemicals, the data evaluation procedure, and so on. Then the scores were given from A to D, just as Miriam mentioned. And here there's an example assay. So we did that for 10 different assays, again, as a pilot to see how it works. So this is the maximum score, and this is what the respective assay got. And then if the sum was close to the maximum possible sum, we coded it in green. And if it was far away, red, and in between, yellow. So this gave a kind of a taste on how does it work with the DNT assays available more or less worldwide. Now, this gives you perhaps a flavor that if we have these 13 domains, then each domain has many, many sub-questions. So there's the test system, what is modeled, how relevant is it, what are the system uncertainties, and that could go like many, many points, and then each sub-item got a point, and altogether there were 10 here. For the main endpoints, there were 4. And just to give you some examples, for instance, for robustness, there are kind of 4 sub-points, so that could give 4 points, test benchmarks, test performance, and so on. So the status is there are lots of criteria available, and they can be used, but for simplification, we just gave 1 point or no point. So what would need to be done to have a acceptable and universally applicable system would be the metrics and the weight of the different points. So this is only an example with the 1, but of course, some points are much more important than others, and that would need to be refined. So then we go to the next step. So the readiness criteria were made with a focus on the scoring. So that would give the readiness status. But then, just as Miriam mentioned, the problem is the information collection for the scoring. Because sometimes it's difficult, sometimes it's nearly unsurmountable to pick all this information from the literature or the test developers. So our solution was a standardized guidance. So ideally a template which has a digital basis, which has then curated information, which can be used for the scoring. And in order to do this, we also took as orientation the guidance document 211, which was also just mentioned in the talk before. But this document is very focused and very short. And this has the problem that it's not structured. It doesn't give exact information on what information is to be supplied. So we more or less translated this document into a form or a sheet which gives much more advice to the test developers. And this was called the ToxTemp template. So it's the template for description of all cell-based toxicological methods. So this is not focused on DNT. And here you see the guidance document 211. It gives a number of items. And we also provided a translation. So the ToxTemp chapter 2.2, 2.3, 2.5 all address this general information. But you see there are many, many more chapters. So there is a more detail and more structuring in this information. So everything in guidance document 211 is there, but it's in many sub-chapters. So this was used in a digital database, and for instance, more than 150 methods from the Tox Risk project were entered there and can be used in a standardized way. And important was that the test developers or those filling this were given guidance, so sub-questions, information, notes on how to fill these fields, because this is not understandable from the original guidance document. So to give you an idea, there are 11 big chapters and they have a number of sub-chapters, so general information, the features of the test system, the features of the endpoints, of test methods, and so on. And to give you more detail, for instance, on point 8, there's a sub-point 8.3. This is how was the prediction model set up. And then there are lots of guiding questions that give those that fill the form advice on how to fill this. So has the prediction model been tested? What was the testing set of chemicals for this? List the chemicals or give the number of chemicals if there are more than 50. So there's a lot of instructions, so this information can be really used for the scoring. Same for test performance, for instance. Just to give you some other examples, outliers were considered, and there's the sub-questions, how are outliers defined, how are they handled, how are they documented, to make sure nothing is forgotten. And this is all available in a digital form, so all these fields and sub-questions are being asked by the computer and are then being collected in a database. So here is an application to the European Tox Risk project. So we thought if there is a test battery and it's to be used in a valid way or in a regulatory way, we need the test documentation and the SOPs and a number of other features. And this is described in a paper where everybody can read the details of how all this was applied to a set of around 25 assays. It's all based on the ToxTemp template, and the database will also be put public so anybody can register methods there. And of course, the background document is also the GIVIMP, so nothing is different from the GIVIMP, it just gives more detailed instructions on it. So if you want to read more, if you want to look after the seminar, so this is all the documentation in the Tox Risk project where you can look it up, and the ToxTemp paper is very clear and gives the whole database of the ToxTemp. Now, another kind of exercise was to use all the DNT assays we could collect and apply a large number of real-life compounds and then apply these basic principles established here. So how do we ideally describe a test method in a way they give enough trust to regulators, and how do we get initial assessments of performance? This is reported here in a large report at EFSA, and at the moment we are also compiling a shorter publication which will be easier to read. So a lot of people were involved here in the discussion, in the planning, in the setting up. And to make it short, a lot of different assays in the area of developmental neurotoxicity were used, and about 120 test compounds were selected. And this allowed for each of the assay to determine the intra-experimental variability, inter-experimental variability, and a lot of performance parameters. All these assays are described in the ToxTemp. They can be found in the OECD database. They went into the OECD projects just to see how would that work in a real life situation. And here, just to give an idea, there were a number of positive controls. So there's a own publication on the selection of positive controls, as mentioned by Miriam, extremely important. And then there's other compounds, there's the distribution of hits in the assays, there's some potency sorting of the hits and double hits and further discussions of this. And in order to estimate at least in a preliminary way, the performance of the test battery. A number of positive controls have been used, a number of negative controls. And then we looked at specific hits, or we included also cytotoxicity or borderline hits. This is why there are 3 different metrics. So I'm focusing on the medium level, that would be the specific hits, including some borderlines. So there are many true positives with the positive controls, but some false negatives. With the negative controls, they are all true negatives, and that gives the performance as indicated here. This gives an initial idea how such a battery could be validated for the readiness, at least for screening. And this is the last slide. I just want to open, like we looked a lot at reliability, to some extent also on the test performance, but of course there are other features to be considered. So this can be reliable, but it's not A+, as Miriam said, it's not the state of the art. And of course, this is a continuing process. So the test system relevance needs to be evaluated. And that is something you can't score so easily. Then, of course, if there are not enough reference compounds, one needs to combine this with a scientific or mechanistic validation. And we did this also in the EFSA report. So we checked for each of the assays which signaling pathways relevant for DNT were represented in each assays and which ones were not represented, and whether we altogether covered the relevant assays for DNT signaling pathways. We compared potency measures to look at the plausibility. Of course, if there are several hits, one needs to check the internal consistency with structure activity and read-across considerations. And of course, it would be good to go into depth, at least with some compounds. So we included 2 case studies where we went into depth for the subgroup, for instance, of flame retardants, and did some further considerations. And of course, if there are hits in one assay, one needs to have a scheme on how one would follow up, or how one would confirm the hits. And of course, toxicokinetics are important if these in vitro data are to be compared to any information in vivo. So I hope this gives an idea on how one can start with the readiness, and then what is to be done for the next 10 or 20 years. And thanks for the attention. And this is a picture here from the University of Constance over the lake and the mountains.
Thank you so much, Marcel, for your very comprehensive presentation. I also posted in the chat some resource that was referenced in your presentation. So we are going to move to our next speaker, Elise Grignard from the Pepper platform. Elise, do you want to share from your screen, or do you want me to share?
We'll try if it's working.
Yeah, it's working. Good.
Okay, good.
Please go ahead, Elise.
Okay, thanks. So just briefly, a little bit of background of about Pepper and what we do here. Pepper is a public-private platform dealing with the pre-validation of methods for the identification of endocrine disruptors. And we are quite a new structure. And we had our first round of selection of methods in September 2020. We do select 3 methods for pre-validation every year. We consider both methods for human health and for ecotox. And we look at methods, in vitro method, but also what we call in vivo light, meaning that we are not planning to embark in a long-term multi-generation rodent study, but we can consider short-term ones. And we only focus on a method that could be used for, and that could fill a regulatory gap. And we do evaluate the method readiness before we submit the methods to a vote for to our relevance committee, who does select 3 every year. So we have three main sources of methods. The first one are databases, for example, DBALM. The second is literature search. So we had a lot of work on defining the keywords for the search. And we also had some help with artificial intelligence to screen the titles and abstracts of the publications. And we also receive submissions directly from test developers. So the first year we selected methods from databases and literature search. The second year we had the three sources. And this year we will have a vote in the next coming weeks. We add methods from literature search and some submissions. So of course, depending on the source of the method, the level of information on the method and so on the readiness of the method is very, very different. So starting with a method coming from publication or from some databases, we start with applying slightly modified test readiness criteria, which has been just presented by Marcel. The main modifications are that we made it endocrine disrupting specific, obviously, and we also made it more applicable to acellular test system and also to potentially in vivo methods. But really the main point for us to have some more knowledge on the readiness of the method that comes from a publication is really discussing with the developer, point which has already been mentioned earlier. That's really the best way to get the information you need. Indeed, in a material and method section of a publication, there's definitely not enough information to make an assessment of the maturity. So the next type of method is the one we receive through submissions. So there's already, if I may say it like that, a positive bias, because if a developer submits a method to whatever structure for the method to be validated, they've already been thinking about it. So quite a lot of work has already been done, at least from an intellectual point of view. And we have put together a form that we ask the submitters to fill in, and we have not reinvented the wheel. It's, again, as already mentioned by the previous speakers, it's based on the OECD guidance document 211 and also on the EURAL-ECVAM test pre-submission form. And what we try to do with that method, obviously to get the answers we need, but also to make the developer ask themselves different type of questions than the ones they ask themselves. And we got this year a submitter who told us that's exactly what happened to them when they filled in this form. And we were very happy to hear that, because that's really what we're trying to do. So here are some of the points or questions we asked the submitters to answer. And you can see on the right that we explain briefly what we mean with the question. So people know what we have in mind, because it's really two different worlds, and sometimes it's not very clear what we might be willing. So we ask them to describe the scientific principle of their method, the intended toxicological use, human health or environment or something else potentially, the purpose of the method, to describe the test system, the tissue, the cells, if there's any metabolic competence of the test system, and to describe the exposure regime. We ask if there's a standard operating procedure, if they have developed some acceptance criteria and technical limitations and strengths of the method. Also some data analysis or potentially a prediction model. If there's any initial assessment of the method performance, has it been transferred to another lab, how many chemicals have been tested, if they have some knowledge about the applicability domain. And we also obviously have some question about the potential regulatory applications with the general context of use, and some specific question, obviously, to endocrine disruptors identification. So the most common issues or difficulties we've been facing when trying to evaluate the readiness of a method. And that's what comes the most often when we discuss with the developer, is that usually the method has been developed for a different purpose. I mean, that it has been developed to answer a specific research question. So the developer needs to change his mindset in order to start answering a question in a regulatory context. We did realize that most of the time the developers have no or a wrong understanding of what validation is. They usually have very low knowledge of regulatory testing, what is needed, what does exist, a type of question which need to be answered. And we also sometimes have to ask them to maybe simplify a bit the method in order for it to be adaptable to some routine use. The most common missing pieces of information, but that you have heard that already with the previous speakers, there's a lack of historical data, very rarely an evaluation of repeatability or reproducibility, because people want to answer a specific research question, they answer that question using their method, and then they move on, and then they also change the next question, so they change a little bit the protocol, so there's no historical data because the protocol keeps changing, and obviously there's no SOP. Very often we found also that there's a very unclear data interpretation procedure, because people very often answer to us, yeah, but I'm used to this data, so I know when something is wrong. So it's more expert judgment, if we may say, than a defined procedure. So obviously that's an issue when you want to transfer the method. And with regard to the prediction model, very often any hit is called positive, and there's no interest for negatives, because they tell us that they don't publish negative results, which is unfortunately true. So to conclude, there's generally a gap between research and validation, is that most of the time the developers have a lack of knowledge or awareness from the developer of what validation is and what is needed to do a validation. And so when the method is not ready enough, you need a lot of preparatory work before the validation, starting the validation, or then you end up with a very, very long validation. And maybe one suggestion would be to develop some trainings, workshops, or whatever tools targeting the developers, a bit what was also presented by Marcel just before, to help them understand what the regulators need. And we have tried such an attempt, not saying that answers all the questions, obviously. We've developed with the University of Paris a web questionnaire, which is called Readiness. So it's for the moment focusing only on in vitro method for the identification of endocrine disruptors, but could be extended further, obviously. And so the questionnaire is divided into several sections. And for each section, the submitter will have a score with a conclusion with regard to that section. Your method is or is not ready to enter pre-validation. And they'll, of course, also have the general score at the end. And so they can keep that just for themselves and we don't know about it. Or if they want more information, they can send the questionnaire to Pepper and we can start a discussion. So that was it for Pepper's point of view, and I thank you for your attention.
Thank you so much, Elise, for sharing your experience with the Pepper platform. Now that you have a few years experience, it was interesting. Moving to our next speaker, Monique Perron from the US EPA. Monique, yeah, excellent. We can see your presentation. You just need to switch to, yeah, good. Give you the floor.
Wonderful. Well, thank you for inviting me. So I'm going to maybe come up a level from the previous speakers, but touch upon many of the same themes. So my name is Monique Perron. I'm a senior science advisor for the Health Effects Division at the Office of Pesticide Programs at the US EPA. And just quickly, just going to provide my disclaimer here that, that the views expressed are my own and may not necessarily represent the views or policies of the agency. And I'm just going to quickly go over this slide. I believe at an earlier webinar, my colleague from EPA, Dr. Anna Lowett, already provided this intro, but I think it's worth reiterating that the underlying principles in OECD GD 34 hold true, but maybe we need to start thinking about rethinking the process to encourage the implementation of what we refer to as new approach methodologies, or NAMs. So any technology or approach or combination of thereof to provide information on chemical or hazard, and that can be in vitro, ex vivo, in silico. So when I'm talking about NAMs, I'm talking about a very broad term. And I think this webinar was sort of timed very well. We just recently co-authored an article in Archives of Toxicology that outlines how we can start moving away from this historical validation process and work on establishing confidence in NAMs. This framework was developed with representatives across various different organizations, so EPA, as well as US Consumer Product Safety Commission, NIH, OECD, as well as a representative from the JRC of the European Commission. So this paper, as I walk through it, focuses on NAMs for assessing human health, but many of the elements are expected to also apply for ecotoxicology. And what this framework did was build off of criteria that's already been developed for evaluating NAMs for skin sensitization that were agreed upon by the ICADM. And also applied the validation principles and that was also applied to the OECD guidance for the defined approaches for skin sensitization. The Casati et al. Document provides that information. Then there's also the framework pulls from the principles in the OECD GD 34 document as well on the acceptance of new or updated test methods. And so when I'm talking about test readiness, it's not just the technical characterization of a test. So we are looking at this from different aspects in terms of there's different essential elements. First is the fitness for purpose. So how is it going to be used? What are you trying to answer, and who are you trying to answer it for? Human biological relevance, as well as the technical characterization that I already mentioned. And then also providing that data integrity and transparency along with independent review. So as you can see in this figure, these elements are interrelated. And so you may have some pieces that will be done separately, but a lot of this will be conducted in parallel. So when it comes to fitness for purpose, we want to understand how will the NAM be used in a scientific context, but also from a regulatory perspective. So what answer are we trying, or what question are we trying to answer? And this may differ also depending on the jurisdiction that you are submitting to. So you need to understand what are the regulatory statutes and what are, what are the confinements of those regulations. And at EPA, we encourage early engagement to ensure that a NAM will provide key information that's needed to make health protective decisions. Those early and consistent discussions also provide opportunities to have discussions to talk with key people. So we're talking about method developers, which has come up quite a bit in the past few presentations, the researchers. We get to pull in our experts on our side who are more familiar with the research and the test methods and can ask a lot of the more nitty-gritty laboratory questions. So it gets to bring everybody into the same room and have those conversations early. And we get to also ask, can you, expand on a particular piece? Can you go back and change a piece of this? So, because right now we don't think it's answering the question. So there's that opportunity for some back and forth and what is and what isn't a potential for a test method. And I would so that basically means that these discussions can facilitate what will be needed and whether or not we can move forward for regulatory use. So again, I mentioned that our paper was focused on human relevance. So the slide is a bit more focused on human, but again, for ecotox, you could step back and think of it as the species of interest. But ultimately, you want to have that understanding of how the test system and the human biology compare to each other. Your confidence is bolstered when your NAM is adequately reflecting the human biological understanding. When possible, it's great to have human data or reference chemicals available. In the pesticide world, that's very limited. But there are, several examples out there where we've been able to, utilize what information's there plus whatever's available from pharma. And then lastly, when applicable, you can evaluate the traditional animal test method in either a quantitative or qualitative capacity to look at that human biological relevance. So we've seen multiple cases where a test species has not been found to be appropriate models for human biology. So this underscores that importance of understanding how well the test method recapitulates the human biology. So technical characterization, obviously we want a study to be conducted in a way that we're getting high quality and robust information to answer the question at hand. We have the OECD guidance, the GIVIMP guidance document that provides, a comprehensive introduction to the elements for technical characterization that should be described in submissions. But they also should provide information, demonstrate data that has been generated using the assays for the question at hand. We also need a sufficient data and information so we can evaluate, the overall, what is the NAM, what protocol are you using, the equipment. So really making sure we have all of that information in a submission for evaluation and consideration. And then also what sort of gets overlooked sometimes is also on the computational side. So when you're using in vitro studies, trying to relate that over to, an external dose, often you're using some sort of dosimetry modeling, which can take several different forms. And so also having that information, the description of the computational models is important for us. And lastly, on this one, I would just say that when we're evaluating the accuracy of a NAM, comparing the data from the traditional animal test, we don't think that that should be the default. And there are numerous publications out there that have recently demonstrated low reproducibility for a range of endpoints in traditional animal tests. So understanding the reproducibility in reasons for observed differences in the traditional animal method allows for setting realistic expectations, not only for the capability of an animal test, so where our bar currently is, but also the ultimate performance capacity. So what you're looking for in a NAM, if we're often trying to set different bars for different types of tests. And I think it provides the ability to have realistic expectations. Data integrity and transparency. So that comes from, the data that's provided. We ask for individual raw data so that we can recreate calculations and, sometimes analyze the data in a way that's different from a submitter. We want to communicate transparently how the NAM is used and the underlying data, and as far as possible, make that information publicly available. And also, we want to assess and describe the uncertainties and limitations associated with the NAM, as well as the traditional animal tests that it's possibly either replacing or adding to, so we can understand how all of the different data relate to each other. And this transparency also, primarily helps us to build trust in the communities and the stakeholders. And with that trust, we hope that we can start to accelerate the acceptance of different NAMs in different capacities. And the last one is independent review. So what is necessary for independent review will be determined on a case-by-case basis by the regulators. Peer review and publication is encouraged, since it can help with sharing information, and it can supplement those more formal reviews that are done by the agency. And in certain cases, this may lead to having a review by an independent third party. So I'm going to just quickly go through an example of this process that the framework wasn't developed at the time, but this example definitely hit across many of the pieces. So in 2014, a registrant for the fungicide chlorthalonil came to us, Syngenta Crop Protection, and said, we have this idea for how to move on from needing more animal data. They had already done some animal data. They knew their chemical was a respiratory irritant for animal welfare reasons, as well as also in hopes of moving to more human relevant information. They proposed this refined inhalation risk assessment approach using an in vitro model coupled with dosimetry modeling. And this was in 2014, they had already put a few years into the development of this proposal, and we recognized the value of it, but we thought there was pieces of it that still needed to be, worked on. And so we worked with them to encourage further development of the approach. We also saw this as a potential approach that could be used for other chemicals, not just chlorthalonil. So we started to collaborate with our colleagues in the Office of Pollution Prevention and Toxics that looks at industrial chemicals, as well as NDP's NICEDM to help with the review. And not on this slide, I accidentally left them off, but also our researchers are in our Office of Research and Development. So starting to, again, bring in that early engagement, bringing all the people in the room. During those early discussions, we were able to, talk with the developers of the in vitro model, who also were the people who were running the tests. We were able to talk to the exposure assessors from the registrant, who also could provide more information about the products. So it's really a great opportunity to bring all the information together. And eventually we, in 2018, convened a scientific advisory panel, since this would be the first time that we would derive a point of departure for inhalation risk assessment from in vitro data. We also wanted to get some perspective on, how far could this be used? Could it go beyond, be used for other contact irritants, as well as other chemicals that have the potential to cause portal of entry effects? So that SAP report came out in 2019, and ultimately no panelists supported us moving back to the laboratory animal study. They did provide some great recommendations on some additional data and information that could be incorporated. And all of that was put into a refined approach that was used for the chlorthalonil risk assessment for registration review. And I'll also mention that after the SAP, Syngenta also published several manuscripts regarding this project. And then more recently, it's also been the basis of an OECD case study. And then just a couple last few slides. I just wanted to mention also that EPA is one of the US federal agencies that's part of ICVAM. ICVAM facilitates the development, validation, and regulatory acceptance of test methods. And in 2008, they published a roadmap for establishing NAMs. And notably, this roadmap identifies the importance of early engagement with test developers, and also encourages agencies to evaluate the relevance of NAMs without relying solely on traditional test methods to define performance. So again, all the pieces are very in line a lot across many of these different roadmaps and ideas of, how can we build confidence in NAMs and start to accelerate the pace of their acceptance. And then lastly, just some ongoing activities that I wanted to mention. So in 2021, EPA put out their NAM work plan. And as part of that, we recognize the need for a framework to evaluate the quality, reliability, and relevance of test methods. So we're currently developing that framework. We also have coming out of that work plan in NIHS, the National Academy of Sciences is conducting a review of validation and scientific confidence frameworks for NAMs. In October, we have a NAM conference coming up where we'll be discussing some of the state of the science for different types of NAMs and approaches. And lastly, ICVAM, we have a validation work group, which several of us from EPA are on, that is working to update a 1997 document on validation. So again, a lot of these components that I've talked through today have really been the same underpinning of several of the different conversations that we've been a part of. And I think I'll go ahead and end there. I thank you again for listening.
Thank you very much, Monique, for this presentation today and mentioning the thinking around, reformulating the validation frameworks. That's certainly of interest also to the WNT. So we are moving to our last speaker for this webinar today, Hajime Kojima, who is going to share with us many years of experience at JACVAM with and the lessons learned from the validation. Hajime?
Yeah. Oh, let's start. Okay. Okay. Can you hear me?
Yes, we can.
Okay. Okay. So thank you very much to welcome, invited me to this webinar. I mentioned that I have a lot of experiment in the variation study and peer review for 17 years. We have more than 20 variation studies and peer review under the ICATM framework, mainly as ECVAM and ICVAM. So until this drawing that we have got a lesson learned at the variation studies. Okay. Sorry. So recently, the sort it's emergency the topics to the MPS to the atoms to model that developed in the worldwide. Japanese correlate is also to develop the 4 MPS devices in the commercialization in the Japanese project. So they hope to start a validation studies. Target organ are liver and intestine and BBB and kidney, and also to do the connection with each target organ. So using this device to test developers to developing the PBPK model for toxicokinetics and so liver and gut models for liver toxicities. So preparing the operation procedure for round robin tests, I think that this is pre-validation studies. So they developed a new type of the PBPK model, MPS models. So I think about the important is the cell criteria. So that this model is to using that to human iPS cells, ES cells, and also to human primary cells. So it's difficult to control whether to anti-give imp, and also to standard for material requirement. It is should be controlled to the quality of the devices, and also to the MPS performance standard to necessary for quality control of the like liver and like kidney. So using this device is to my colleague to developing the standard operating procedure to each toxicokinetics and also to the liver toxicity and also to kidney toxicities. So they hope to just start the validation studies, but so it's difficult to start the validation study because the validation studies are very simple and not simple and so easy. So test and also to test developer, mainly the academia has many concerns with the validation studies. They mentioned to me that my protocol has already published, and so also a complicated process on the guidance document 34, and so excessive demand is not acceptable. Long-term restraint not acceptable. They have no budget, and also after the validation study, why not a peer review? It's difficult to communicate with the sort of academia, and also for test guideline, they cannot so discussion in English. Maybe so almost Japanese. It's not easy to discuss in the English. So we have a lot of concern with the validation studies. However, so we have to develop a simple and easy validation studies. So evaluation criteria for validation novel test method. It's important. It's a subject, object, study object, and biological mechanism relevant, and so protocol, and so chemical selection and reference data. So previous speakers mentioned. And also to transferability and within between laboratory reproducibility, predict capacity with applicability domain limitation. We have to solve the step by steps. So I consider that simple validation studies. Validation study and novel test method are crucial for regulatory acceptance of new approach method. But limitation is numerous evaluation criteria before acceptance and the difficulty in performing an international study owing to limited budget. Members involved become exhausted while performing the several phases owing to device immature protocol. And the availability of pure database concerning several fields of toxicology. So because Japan is no database such as NTP and ESETOC. So this is my proposal based on the lesson learned. So detailed review paper including the performance standards, absolutely including the reference chemical, should be developed with each systematic toxicology by OECD or the other organization. And international experts who are familiar with test guideline development are mandatory on the validation management team and the peer review panel. The purpose of the study is limited to checking the robustness and reliability of the protocol based on the performance standard. Mainly, I think that the acceptable chemical number is up to 30. So the bottleneck validation study is resolved and simple and easy. This is a case study of some validation studies. This is case of the epicenter for skin sensitization. That these assays are acceptable to SPSF and also including the work plan. So this validation study starts to 2018 or after the transferabilities. And so to start May 2018, almost 2 years under the COVID-19 pandemics. In Japan, these laboratories are closed to the COVID-19 pandemics. However, so my colleagues are to validate this to each phase. So one phase is total is 5 chemicals is this phase, and 1A is 5, 1B to 5, 1C is also 5 chemical use. So for within and between laboratory reproducibility, 3 times each. So for example, that one laboratory is to validate to the 15 chemicals to each phase. Anyway, so to VMT members discuss after phase each phases. And so in this process, VMT members to revise the SOP, so 3 times. And so very easy between laboratory reproducibility is a phase 2 studies. I think that this phase is 2 months. So experiment part completed in March 2020. So total is almost 2 years completed the experiment part with the validation studies. So I think that this validation study is a performance standard based on Ekman validation study. You know that I say again that I have a lot of the experiment with the validation study, but I prefer the Ekman skin sensitization validation studies. So I think that this is good study plan. However, this validation study is very complicated. But so based on the Ekman experiment, we have to consider the simple and easy study plan. And so predicted capacity and applicability domains based on the in-house data after the validation studies. So observatory including the discussion is a predicted capacity in the validation report. So now is that ongoing the peer review with these studies too. But so it is good cases to the validation studies. I think that the total 1 or 2 years in experiment part is acceptable for our test developers. However, so long period validation studies are not acceptable because the budget is limited and also to manpower is insufficient. A shortcut of evaluation criteria is important. Checkpoint prior to the validation studies, object and so need a benefit and so biological mechanical relevance and protocol. Appropriate validation study management team, mandatory of the high quality of validation management team. And so mission during the validation studies and check of the transferability and so quality assurance system, and so within and between laboratory reproducibility, and also to completeness of the data and documentation. This is a mission with the validation management team, but I think the important is a checkpoint or the prior to the validation studies is it's crucial. Okay, so for non-validation studies, I think the test method is an important element for NAN. The purpose of it is limited to checking the robustness and reliability of the test protocol. Before starting the validation studies, I think the important is that before starting validation studies, the VMT members should be discussed the object, benefit, and biological and mechanical relevant with the test developer and the study plan, including the acceptance criteria, absolutely including an application model with the sponsors. VMT members should discuss the protocol development and chemical selection during the ongoing validation study. However, if a detailed review paper is developed in the before validation studies, the mission of the VMT member is to only develop the protocol. So in this case, is that acceptable is a test guideline. So I think that to propose to you that this simple and easy validation studies for now. Okay, thank you for attention. This is also building with the NIHS in Japan. Thank you so much. I appreciate you.
Thank you, Hajime, for concluding the last presentation of this webinar today. I see that there has been some exchange in the chat, and thank you for that. This will not be lost in the short webinar report. And This short webinar report will be prepared shortly and posted on the WNT website. I thank all speakers today for their time and commitment. I would like to mention that we have just set some dates for the upcoming 2 webinars, webinar 4.1 and 4.2. They will touch upon reproducibility issues, and they will be on 23rd and 27th of September. The invitation will follow shortly. I know you wanted to have some dates, so at least those can be marked in your calendars. And well, without further ado, I wish you all a very good day, good evening, and we'll talk to you soon again. Bye.